This video is going to discuss transport. No, not transport across the universe, but transport in and out of cells through the cell membrane. Objectives for today are to discuss the structure and function of the membranes of living organisms, because that is what things are transported through, and how materials are exchanged across these membranes. So to start off with, we have to look at what a cell needs, because that is the whole purpose of things being transported in and out of a cell. So the basics of what a cell needs is water, a correct balance of ions, nutrients like sugars and amino acids in order to maintain life, and some need hormones. And they also need waste to leave when it is created within the cell. So it all goes through your cell membrane, which is known as a fluid mosaic model because it keeps moving, which allows things to move in and out of it. It is, as we learned with the cell and organelles, it is a phospholipid bilayer with proteins that are embedded in it, and it regulates what goes in and out of your cell by being selectively permeable. So things that can go through are generally just small, good rule of thumb, and also need to be hydrophobic and or uncharged polar molecules. So examples are oxygen, carbon dioxide, which are important because those things need to easily move in and out of the cell, and water, glycerol, and ethanol. Things that cannot go through, or at least can't go through with the help of your proteins, are larger uncharged polar molecules such as amino acids, glucose, nucleotides, and ions, so any charged molecules are not able to go through without the help of transport proteins. And how things actually move in and out are through a few different types of transport depending on the type of molecule. So you have passive transport, which is transport with no energy necessary, and three different types are diffusion, osmosis, and facilitated diffusion. So note, passive is no energy. Then there's active transport, which, as you see in the picture, needs energy to move things through the membrane. And then there's endocytosis and exocytosis, which are basically opposites of each other. So to start off with, diffusion is the movement of molecules from high to low concentration. So as you see in the picture, the top one, you have molecules of dye on the left side, and they will diffuse across that membrane until equilibrium is reached. So note the area of high concentration towards the area of low concentration. And even with two solutes, they will move from their area of high concentration to their area of low concentration until equilibrium is met. So all of these things like oxygen and carbon dioxide are able to passively move across the membrane, will always go from high to low concentration. So as this says, it is always due to a concentration gradient. A general example is how smell permeates a room. Osmosis is basically diffusion with water, and it gets slightly confusing because generally people think of concentration in terms of the solute, not the solvent. So when you think of high to low, it is moving from an area of high water concentration to an area of low water concentration, but often it looks opposite, so beware. And there are two types of solutions when discussing osmosis. There's hypotonic and hypertonic. And the way I can remember them is hypertonic is when discussing a cell, the water wants to get out. So water leaves your cell and moves to the outside. And if you think of when you're hyper, you have lots of energy and want to get outside. And hypotonic is the opposite. So these pictures show a solution of hypotonic on the top, and you'll see that there's 20% salt solution on the inside of the cell and 10 out. So the water wants to move into the cell. And you can also think of it as where salt goes, so goes water. So in the other picture, you have a solution that is hypertonic and water is moving out. And there's also the isotonic, which has equal amounts, so water moves in both directions and it's not moving more in or out. An example of these osmotic solutions is found in a plant cell. And you can actually see the plant being wilted, and that is because it's going through plasmolysis, and when it's bright and cheery, it is turgor pressure because it has more water. So if you notice in the grocery stores, they constantly spray water on the lettuce in order to keep it crisp. Due to the turgor pressure of the lettuce being in a hypotonic solution where the water is going into the cell, causing the cell to swell and there to be pressure against the cell wall. And now on to types of transport that use proteins instead of just diffusing through the membrane. Facilitated diffusion is still a type of passive transport, but it just uses transport proteins. Active transport uses proteins and requires energy, so we'll get a little more specific into that. Facilitated diffusion is used for larger molecules and all ions. So ions have these things called ion channels, and those are your transport proteins, and each channel is for a specific ion. So you can have a sodium ion channel or a potassium ion channel, and they still move from an area of high concentration to low. They just need the transport protein to go through. So active transport is in general just transport when energy is needed, because diffusion can't be used for everything. If you remember back, not everything just easily moves through the membrane. So an example of active transport is when a sodium-potassium pump actually pumps sodium and potassium against the concentration gradient. So as you see in the picture, 3 sodiums are leaving for every 2 potassiums to come in, and it actually creates an electric gradient. It's very important in your nerve cell, but it creates a positive outside and a less positive inside, because the 3 out to the 2 in. And because it's moving against the concentration gradient, facilitated diffusion cannot work, so it needs to use active transport, which uses energy to literally pump these out. It's kind of like instead of going down the slide with ease, you have to work your way back up the slide. And for our final types of endo and exocytosis are for extremely large molecules, and basically the cell membrane folds around the substance to transport it in. And endocytosis is when it comes in, and exo is when it's leaving. So this shows the top is endocytosis, and the molecules are being pinched, and it creates the vesicle inside, and exocytosis is when things are leaving, and that's generally how waste leaves. So as you see, this is how it is used in your cell. When you are making something that needs to be secreted, your exocytosis is used. But when you actually need something that's very large, like a lot of unicellular organisms, eat through this way, and you can get a large molecule in, or a large substance into your cell through endocytosis. So to end it with the general purpose of transport in and out of the cell is to get the cell what it needs and get rid of what it doesn't need. So all the oxygen and carbon dioxide and materials that it needs, like nutrients, come in very easily, and waste materials exit through all these different types of transport, passive and active as general, and endocytosis and exocytosis.